Hello, everybody. My name is Jennifer Martin. I'm the new editor of the Pharmacology Research and Perspectives Journal. We're here today because it's actually 10 years since the first publication of this journal as a gold open access journal collaboration between the American Society for Pharmacology and Experimental Therapeutics, the British Pharmacology Society, and Wiley. Today is our 10-year anniversary of that journal. And since its inception over 10 years ago, the journal has really flourished in terms of the numbers of papers submitted and published and the increase in impact factor. We've always encouraged people to submit work directly to us, particularly around the education theme, but we've also welcomed manuscript transfers. The sort of areas that our journal covers, as well as being a cascade journal from both the British and the US pharmacology societies, are we're really trying to encourage submissions of original research, but we've also considered and published short reports, reviews, and topical perspectives that address key issues in pharmacology. These include basic pharmacology and clinical pharmacology, and we have an editorial board and review team that covers those aspects of pharmacology. We do really encourage people to review the author guidelines when they submit to our journal, because there are some areas of the journal that we don't focus on, including complementary therapies and some other aspects of more pharmaceutical design. Our first impact factor was in 2019. Our impact factor was 2.05, and we've increased to 2.97 for 2022, and a slight dip, as all the pharmacology journals did, for 2023. We're really, really keen to hear from young researchers, and we encourage you to look at our journal both for a direct submission and also as a cascade journal from both the British Pharmacology Society and from ASPET. I'm a clinical pharmacologist and I practice as a general physician and a clinical pharmacologist. And I'm really delighted to be working with Ross Corridon, whom I'll hand over to in a minute as our deputy editor, and whom will become the editor-in-chief of this journal in a few years' time. I'm also really grateful to have taken on the journal after a really significant leadership achievement of Dr. Mike Jarvis, who led the journal in the 3 years prior to 2024 and did an outstanding job of trying to encourage good quality research from members of both societies, as well as being aware of the significant changes in pharmacology research globally. More recently, we've really tried to focus on getting submissions from areas of the world that haven't traditionally been strong submitters to our journal, Pharmacology Research and Perspectives. And we've noted that there's been a significant increase in submissions from countries like India, from South America, and from Africa. And you'll see when you look at our editorial board that we've increased the numbers of editors and reviewing editors across those countries. We're really delighted that British Pharmacology Society has agreed to host this webinar today. We think it really focuses on the key issues in pharmacology at the moment. We're really delighted that we've reached out to people who are experienced in pharmacology and also people that are beginning their research in pharmacology, both from an academic and a clinical perspective, but also from our key pharmacology researchers in the pharmaceutical area. So with that, I might just hand over to my colleague, Deputy Editor, Dr. Ross Corridon, and then we'll be formally opening the webinar. Thank you.
All right, thank you very much, Jen, for the introduction. So my name is Ross Corridon. I am the new Deputy Editor of Pharmacology Research and Perspectives. Thanks, everybody, for calling into the meeting today. I know we don't have a whole lot of time, so I just want to say two things. We very strongly encourage submissions from early career researchers to Pharmacology Research and Perspectives, as Jen said. These can be review articles based on a thesis chapter. We also accept replication studies or negative results. So, please consider us when you're preparing a manuscript. Then finally, please feel free to reach out if you're interested in contributing as a reviewer for the journal. So we're looking to build our editorial board in particular. So send an email to either Jen or I if you're interested in participating. Thank you very much. I'll go ahead and hand it over to the next speaker.
I think that's to you, Sam. Yep.
Yes, good morning, everyone. So thank you for joining us today. I'm incredibly excited to be part of this 10-year celebration for PR&P. And I wanted to say a huge thank you to all of the organizers for making this happen, and welcome to you, the audience. We've got an incredible lineup of speakers, ranging from people who have had success in academia and or industry to those who are just starting their career in research. But before we hear from those inspirational journeys, I just need to make you aware of some housekeeping. So each of our speakers will be given 13 minutes to present. And after that time, we'll give 2 minutes for questions. We'll then join together at the end for a panel session where more general questions can be asked to each of our speakers. If you wish to ask questions, at the bottom of your screen, you'll see that there is a Q&A function. Please only submit your questions via the Q&A function, as the chat function will not be closely monitored. Please ensure to mention with all your questions who you would like that question to be directed to, because at the end of the talks, questions may not be answered at that particular time, but we can reuse those questions in the panel discussion. Okay, and then one final thing to make note of is that this webinar will finish at 11:30 and the connection will be terminated at 11:30. So thank you again for joining us. I hope you find these talks both interesting and inspiring. Now I'd like to hand over to Dr. Eddie Ragg for the introduction of our first speaker.
Hi there, thank you, Sam. I've just got a few more to add there. So just notify everyone that this meeting is recorded and it will be made accessible after the event. And if any member of the audience encounters any technical difficulties, please ask in the chat. And if you're not in the meeting, then please email meetings@bps.ac.uk. Right then, let's start with our first speaker. So our first speaker today is Dr. Nicola Dijon. And so Nikki is a research scientist at Sosei Heptares. And Nikki, I've got to say, I did have to reread the title of your talk a few times, but it is GPCR, Drug Discovery, from Academia to Industry to Academia to Industry. Over to you, Nikki. Thank you.
Sorry about the mouthful. All right, let's get this loaded up. All right, so I will assume everyone can see it, not in the right view, of course, because that'd be too easy, but there we go. Right, so thank you very much for the organisers and congratulations to PR&P for their 10-year celebration. So today I'm going to talk about I feel kind of a bit of a fraud. I've only been kind of in GPCR drug discovery for less than 10 years, but kind of what has led me to where I am today. Back in September 2013, I came to the University of Nottingham to do a 4-year integrated Masters of Neuroscience. Whilst I was there, I met a lecturer called Nick Holliday, who ended up really informing my career and being a fantastic mentor. So I came up to him and said, molecular pharmacology sounds really interesting. How can I get more involved? And at the time, he had a PhD student, Lisa Stott, and they let me come into the lab and see what was going on, what research in an actual job, not just in textbooks, really looked like. So I was able to do a summer placement with them in 2015 and really kind of get to grasp of, is this something I wanted to continue my career in? At the same time, because it was an integrated master's project, I spent 12 months at Janssen Pharmaceutica in Belgium. Now, I was doing in vivo neuroscience research there, and one of the things that really came across was this very large organization. It was set across Belgium and America, and there were other sites. So everyone kind of had set rules. It was a very structured organization. but it was a really good insight into kind of how big pharma works. The other thing is I lived in Belgium for a year. It was fantastic. Honestly, one of the best experiences I've ever had. And, you know, it really kind of gave a real kind of deep dive into the culture, and I really enjoyed it. So after finishing up my undergraduate, I decided, yep, research science is for me. And I went back to Nick Holliday and also Stephen Charlton and said, I would like to do a PhD with you. However, before we even got the funding, Stephen Charlton had recently spun out Accelerate Bioscience in Nottingham. So this is a contract research organisation. And at the time, it had less than 10 people, and it was still working out of labs based at the university. So I came on for a couple of months, kind of as an extra pair of hands, but I learned so much in that experience. So it was a very small organization. You had multiple roles. At that time, they were moving from the university to kind of an independent site. So there were things like you're moving these pieces of equipment which are hundreds of thousands of pounds, and you know, you kind of learn that even at that stage you can have those responsibilities, and it was a great opportunity. Also, during my PhD, I went and did a 3-month placement. So this was at OMass Therapeutics. So my PhD is all about molecular pharmacology. So this gave an insight into a new topic. So this was a kind of medium-sized organization. At the time, they were actually based on 2 sites, both in Nottingham and Oxford. They've now kind of combined. But at the time, I was in the Oxford site, and there was definitely logistical kind of challenges with working within 2 sites, and it was interesting to see how they got around that and everything. And I had to adapt to these new roles. So I wanted to learn about protein engineering and native mass spec, which I was kind of aware of, had vague kind of experiences here and there, but I'd not done like a proper 3-month placement. So again, it was being thrown in at the deep end, and it was this was kind of the time where I was like, yes, industry, drug discovery, this is really for me, and these experiences are all starting to kind of add up. Just on one personal note, you might have noticed it was February 21 to April 21. UK was in lockdown. So during this time, I was living on my own in an Airbnb. Going to work was my socializing. So when you think about, do I want to move somewhere? Do I want something new? Don't do it maybe in lockdown, and make sure you've thought about the kind of personal implications. So I'll go into the data of what I've kind of a story from my PhD. But first, I just wanted to say PhDs are great for networking. You go to conferences, you meet people at kind of webinars like this and say, hi, I saw your talk, it was really interesting. And I have met people from during my PhD who I'll be friends with forever, who I'll be colleagues with. And, it's a really good opportunity. And I just wanted to say thank you to everyone who was a part of that. So as I said, I will have a quick bit of data. We're limited on time, so I'll just take kind of a short story. Now, I don't know what everyone's backgrounds are, but I worked on something called a GPCR, so G protein coupled receptors. They span the membrane and signal between extracellular and intracellular compartments when at the plasma membrane. They can be activated by agonists, blocked by antagonists, and they can signal through multiple intracellular pathways. Now, this is very interesting. Can we exploit this, prefer one pathway over another? I'm not going to go into bias because that is a whole kettle of fish, which I refuse to touch. So one of the questions we were asking during my PhD was, can we figure out how different ligands bind the receptor if it's coupled to a specific intracellular effector protein? So we took the most basic receptor of all, the beta-2 adrenoceptor, and we pre-coupled it with the mini-GS protein. So this is its canonical, synthetic form of its canonical binding partner, and we stabilized this with nanobit fragments. We then did a TR-FRET binding assay, and I'm not going to go into the kind of minutiae of it, but just so you know, we kind of tagged the receptor, and if a fluorescent ligand was in the proximity of the receptor, you got transfer of energy, and this was an indication that the ligand was bound to the receptor. So our question was, do agonists and antagonists bind a receptor the same when a G protein is bound? So we either had our receptor on its own or receptor with the mini G. So this is a lot of data points, but effectively all you need to know is as you increase the amount of fluorescent ligands, you get more and more of our TR-FRET signal. And that means there's more and more binding, which is logical. We set this to a mathematical model that tells us how quickly our fluorescent ligand binds, the K on, how quickly it comes off, the K off, and the resonance time, so how long it's actually in the receptor. We can then take a kinetic KD, which is an output of how well it binds the receptor. Now, what you can notice here is these figures aren't that different, and this isn't that surprising. So the fluorescent ligand we used had an antagonist pharmacophore, which means it doesn't really prefer the active over the inactive, and there doesn't seem to be any difference kinetically. So now we can go on and add unlabeled ligands, and we can see how they differ in their binding. So what we did is we took two full agonists, so isoprenolone and formoterol, so iso and 4. We took a partial agonist, so salmeterol, and another antagonist, ICI 118551. So you can see the blue bars here are where we've added the mini G protein. So the graph on the left, you can see that the association rate, the rate at which our ligands are binding the receptor doesn't seem to change. However, on the graph on the right, you can see what it is that is governing this shift in affinity is actually dissociation rate. So once these ligands bind the receptor, the full agonists, isoprenolone and formoterol, leave the receptor much, much slower. Salmeterol leaves the receptor quite a bit slower, and ICI or antagonist doesn't. So our conclusions were that our agonists show increased affinity, and this is driven by slower dissociation rates, and that the antagonists show no preference. So my question was, this is great, it's great data, but I've spent so much time in industry, how could we use this in a drug discovery setting? So the idea is if you pre-couple your receptors to intracellular pathways of interest, ones that you know trigger internalization, desensitization, potentially you could identify specific ligands that may activate those pathways over others. Right, so that was the kind of roundup, very quick little snapshot of data. And what I really wanted to show was that even though I was doing academic research, I was still interested in how this could be applied in a drug discovery setting. So, right, so I finished my PhD. I'm looking for a postdoc or an industry position. And I was working with Nick and Steve at the time, and they said this project's come across we've been working on, which is a DPFS MRC project, which effectively says, here is a beautiful project that is working towards a product that can be commercialized at the end. So I was working with Nick and Steve and Shailesh Mistry on an idiopathic pulmonary fibrosis project. So just to kind of sum up all the reasons I think it was a great project, basically. So I got to stay in GPCR pharmacology. This was an area that I've been working on for quite a while, and I'm getting more and more interested in. Excuse me. And I really wanted to stay with it. And also the people that surrounded me within the University of Nottingham were so well informed. I got to work in a drug discovery project. So I have been eyeing up industry positions at this point. So I really wanted something that had a drug discovery kind of flair to it. So I liked how this project had a specific workflow and a pace it needed to keep up. It also had milestone requirements. If at month 6 or month 9 we hadn't reached those, the project could have been kiboshed and we could have not continued. That didn't happen. And it really was kind of a crash course in a lead optimization program for me. I learned an incredible amount during this 2 years. Practically, I learned lots of new disciplines. So I had done lots of recombinant work. I'd done some protein work. I did mass spec, but I'd never done any primary cell culture or phenotypic assays. I went on to do regular screening and also target engagement studies of in vivo samples from our collaborators. One thing that's not spoken about is I did quite a lot of compound management, and this is always something you have to consider. Where are your compounds? How much is left? Are they degrading? And it was a really important task. And finally, what could kill a lead? So there were many things, for example, for the pharmacokinetics, were these drugs toxic? All things like that. Personally, it was really good for me as well because I hadn't actually submitted my thesis at this point. So I could write very late into the night and then submit it. And because I was within a close proximity of my PhD supervisors, that suited. And I worked with really fantastic people, like really great people. I've just put on a couple. There were so many more that worked on the project, but this really led me to having like a fantastic 2 years where I really learned a lot. So once that finished, what comes next? So at this point, I had eyed up a lot of different places and got a few interviews. So behind door number one, I got an offer from a prestigious academic institute who were trying to do, they were doing drug discovery within an academic kind of environment. But this was not solely GPCR research, it was switching more back to neuroscience. And the career progression, the only reason I put a question mark, it was a fantastic position. The people seemed lovely. I was really excited, but I had always seen myself moving to industry at the end. So was I going to delay where I was going to be in 5 years if I took this position? Behind door number two was a well-recognized pharmaceutical company that had like international recognition. They did GPCR drug discovery and an industry setting at a really good pace and a good breadth of projects, and it had the prospect of having really good career progression. So I took door number two, and that's led me to where I am today. So my role at Sosei Heptares is a research scientist where I do GPCR drug discovery. I do assay development and screening and working with multidisciplinary teams. And again, I find myself very lucky. I've worked with some wonderful people, and I still do today. Right? So that's just a little show of where my kind of different experiences have taken me and how they've informed me. Yeah, so that's about it. Any questions?
Thank you very much, Nikki. So we'll just open up the Q&A, and there is a question for us right now. So, hello. Firstly, thank you for the talk. I may have missed the part, but did you apply to Sosei Heptares internship? And if so, would you be able to direct me to something similar at Sosei if it still has something like this? Thank you again.
Yeah. So I didn't do an internship there. They do academic internships. They run, so if you wanted pharmacology, I believe there's 2 placements per year, but they are decided quite ahead of time. So we've currently got 2 students in, and they've already decided on the 2 students who will be starting next September through the academic year. So I'm not quite sure when they advertise, but for the next year, but they'll be there eventually. I applied through Sosei Heptares to do a job. I saw it advertised and I applied for it, and I just did it the regular way.
Fantastic. Thank you, Nikki. So I'll just take one more question and then we'll move on, but there'll be plenty of time in the panel discussion to ask any more questions for Nikki. So Nikki, do you have any tips on finding mentors in this field, as it seems that that's benefited you a lot?
Yeah, I think you have to look at what you find really important. So do you want someone who has worked in industry before? They could tell you good places to go, good people to meet. Or if you want to stay in academia, do you look for someone who has a really good publication record? They're different things to look for. You need to find what you want to do and find someone who's maybe had a similar journey or potentially has similar contacts or useful contacts. I just look for people that seem to have done things I would like to do, basically.
Sounds a good idea. Right then, I'll pass on now to Sam to introduce our next speaker.
Thank you, Eddie. So our next speaker is Dr. Chloe Peach, and she'll be telling us of her story of how she's reached academia and some of the research that she's done along the way.
Thank you for the introduction, and thank you very much to the organisers for the invitation to speak today. So I've given you a bit of a spoiler in the talk. So I'm going to talk to you about my journey, essentially, going from the UK to the US and back again. I also noticed that a lot of us put that in our titles, so you might be seeing a lot of journeys like that today. And going through some of the science that kind of led me into this direction. So to start right at the beginning of this journey, so during school, I actually didn't necessarily love biology that much. I was more of a math kind of student. But then I remember during A-levels, a particular lesson where they started describing proteins and the 3D structures of proteins. And I got this image of my head, and that's kind of what started my love for biology. So then, because of this interest in biology, when I was thinking about applying to university, I wanted to essentially know how the body works and the science behind medicine. So logically, should I become a medical doctor? So I did actually apply for medicine originally. And then I went to my first interview and realized that I potentially wasn't doing this for the right reasons. It was that I wanted to think about the science behind the medicines themselves. So I changed my mind at that point after that first interview, following that gut feeling that essentially what I wanted to do wasn't medicine per se, it was the science behind medicine. So I chose to do neuroscience instead. So at this point, so I'm also going to put in a couple of tips as I go through and then some more at the end. So this would be to follow what you're actually excited by, scientifically, etc. So at this point, I then moved to Nottingham to do my neuroscience degree. And again, following that gut feeling. So actually a lot of the modules that I enjoyed in particular were the pharmacology modules, the biochemistry modules, etc. And then during a seminar with Jeanette Willard, who actually helped organize this session, she was talking about some of her work looking at growth factors and in particular how tumors themselves can grow their own blood supply, and how she found these really cool new side effects to a drug that you couldn't actually see unless you looked at them in this specific method. So purely essentially because I was super interested in what she showed us, I then reached out to her. And then that led to a summer studentship working in her lab and in the Nottingham lab here, funded by the BPS. And unlike some of the labs that I had done during my undergrad, this actually gave me a real insight into what it's actually like to work in a lab. So firstly, that labs have a window, they have a radio, and you get to design your experiments independently. So it was essentially really fun, and I didn't really expect it to be quite so fun. And then the thing that really hooked me in, and I was then definitely going to do a PhD after this, was attending my first conference and also presenting my first poster at a conference, where you essentially get to talk about all the science that you're excited by and with the experts themselves. So my second tip, even at this stage, talk about the science that you're excited by. Reach out to those professors that are talking about really interesting things. Even if you feel like you don't know anything, you're probably going to ask them really fun questions and bring your own perspective. So then thinking about the actual science that drove me. So I mentioned that Jeanette Willard worked on growth factors. So what we're really interested in are these receptor tyrosine kinases. So this is a single transmembrane protein that acts as a dimer and interacts with these growth factors. This then leads to a phosphorylation and intracellular signaling pathways that leads to things like proliferation, survival, and migration. And a lot of the pharmacology fields don't necessarily look at receptor tyrosine kinases. There are certain drugs that target both the growth factors and also RTKs themselves with these RTK inhibitors, but they're not necessarily looked at beyond the field of oncology. And in particular, when we think of how receptors actually work, they don't just act at this end point after 60 minutes. They're actually very dynamic, and not all biology is very dynamic. So kind of going back to that image of how biology is actually working in our head, it's these dynamics that I was really interested by. So this, at the end of my bachelor's, I then went straight into a PhD, and this was funded by the BPS as well, by an AJ Clark scholarship. So this was following on from my summer studentship project, where we were looking at the vascular endothelial growth factor and the VEGF receptor 2, so this RTK that leads to angiogenesis. And in particular, we were looking at how this co-receptor called neuropilin can modulate its activity. So you might recognize this word from my title. So essentially it can upregulate RTK signaling, and it does so through interacting with the ligand and also potentially through indirectly acting with the cytoskeleton. But in my head, we don't really know how it actually upregulates its signaling. So this was, again, following that scientific gut feeling of, this is really interesting, and I can't figure out how this is working from the data that there is. So one thing I'd also point out here is the importance of mentorship, and that's very much mirroring what Nikki said as well. throughout every career stage, and having a whole range of people that you go to for mentorship. So particularly my mentorship team during my PhD, for example, with Jeanette, Steve, and Laura, it can really make a difference to not just supporting you, but also challenging you and kind of prodding you when you feel like you can take an extra challenge. So just to briefly show some data. So the love for neuropilin begins. So essentially we wanted to tease out some of the molecular pharmacology of the VEGF receptor compared to the neuropilin co-receptor. And essentially we used bioluminescence resonance energy transfer. I'm not necessarily going to go into that. I'm just going to talk through what we actually found. So when you look at the KD, that binding affinity, they essentially look quite similar. But when you look at these dynamics, they're actually quite different in how they interact with VEGF. So going on to think about how neuropilin actually upregulates RTK signaling, this led us to think that surely this must mean that neuropilin speeds up RTK binding. So you have this RTK binding, which is a lot slower, and then the interaction with this co-receptor is much quicker. So surely we've answered it. We figured it out, the question of how neuropilin actually works. But actually, it doesn't necessarily do that. So our answer has not necessarily come to its fruition yet. So we can see that when you look at that interaction with both VEGFR2 and neuropilin in a complex, it hasn't sped up RTK binding. So the question still continues, how does neuropilin actually enhance RTK signaling? One thing I would point out here is that I was definitely very lucky to publish quite a lot from my PhD. But sometimes obviously that can be down to good or bad timing. So just think about other ways to boost an academic CV, or thinking about the kind of journals that you can publish in. So now for chapter 2. So at this point, I'd finished my PhD and I knew that I wanted to move continent. I don't know why, but I just wanted the personal challenge of moving continent to see a new culture, to experience things. And I do note that, again, not everyone has this luxury, not everyone can make this decision, but at this point I was able to do so. And I wanted to move somewhere English speaking because I can't speak any other languages, unfortunately. So US, Canada, Australia. During my PhD, I met with Nigel Bunnett, who was based in the US, and I met him at a number of different conferences. And we got on really well. So I loved his work because endosomal signaling is essentially really cool. So like I was saying about following that gut feeling, it would involve a shift in the field. So going from working with angiogenesis to the field of pain, and also shifting from my evident love of RTKs to GPCRs. So his work was really cool. So even though it was changing to a new field, going from a very small fish to a very big pond, especially thinking of the US as well, it's a challenge, but the perspective that comes with it is definitely worthwhile if you're able to make that decision. So one thing I will point out with respect to the journey not always quite going as planned. So when I had the interview in New York and accepted the job, it was actually 2018. So I was in the first or second year of my PhD, which is quite an early time to arrange a postdoc. I don't think many people arrange it that early. I then accidentally met someone in the UK. So obviously life's starting to get in the way a little bit. And then not to mention the fact that I was meant to move in, I think it was April 2020. So obviously it didn't quite go to plan in that sense, particularly because the US introduced this travel ban with Europe. But we did eventually make it there. I think I was delayed by about 4 months. I had to get an exemption so that I was actually able to enter the US despite this travel ban. But I made it there. This was actually a picture of me on the way from the airport to my new flat. You'll see face masks and you'll see moving overseas with just 2 suitcases. But it was a great challenge. And it really makes you appreciate how challenging it must be for other people that have also moved to the UK with respect to visas, with respect to moving with a suitcase. And it's something you just don't really consider until you actually do it. So then to give you some impressions of what it was like working in the US, I don't know if Ross is still on. It was amazing, let's be honest. It was so collaborative. The amount of people working on projects was incredible. Therefore, the pace that it can go at is ridiculous. Obviously, there's a lot of money there, which helps that. There are also some changes just in the way everything's managed. So PhD programs themselves are up to 7 years and even above, because there's not as much of a time limit as there is elsewhere in the world. There were differences in how labs are run. So there was no one ordering things. I was basically just loaded with everything and got to manage it myself, which I ended up loving because I love the organization. But it's just a different way of working. Also, we go from working on 1, maybe 2 projects in your PhD to there within a few months, I was working on maybe 6 projects plus. And obviously that shift in work-life balance, which is very lab dependent, but I'd say as a whole, obviously the US is very work oriented. What it's actually like to live there as well, to give you the kind of pros and the cons. So obviously I moved during COVID, so a lot of these are linked to COVID. So there were no in-person appointments, visas included, there were no flights. Obviously with a long distance relationship, that was horrible. But this isn't necessarily COVID related. I didn't find out that there wouldn't be furniture in the flat until about 2 days before going to the US. So that was fun with a 2-week quarantine. But there are obviously a lot of benefits. I was there for 3 years and I absolutely loved it. So the people that I met, New York as a whole is amazing. New York as a whole during COVID meant there were very few tourists. So I basically got to explore the entire city without having to get in the way of anyone. And it was just an amazing experience. So just as a very quick overview of then how I shifted that research focus. So obviously I said about shifting from angiogenesis to pain. And at this time, the Pain Research Center was established at NYU. So I worked with GPCRs for, let's say, the first 2 years primarily. But I was starting to look into this field of pain. And again, that gut feeling of growth factors and receptor tyrosine kinases. There is a very key growth factor that's involved in pain, to the extent that if patients have a mutation in this protein, then they actually can't detect pain at all. And it acts through this receptor tyrosine kinase track A. And I won't go into too much detail about how it works, but it essentially is a growth factor that causes pain. When I actually looked at the sequence of NGS, I noticed that there were a few binding motifs that could actually interact with neuropilin, the protein that I keep seeming to be obsessed by. And in order to investigate this particular project, I was awarded a fellowship in neuroscience by the Leon Levy Foundation to embark on this project. So it was a postdoctoral fellowship, but kind of going towards independence. And I should also note, this is something I wanted to point out for that kind of Instagram versus reality, what you see versus what actually happened. This was my 5th application. So I got a lot of no's, a lot of definitely not, a lot of 2% success rate no's, but keep going and don't always just look at what you see on Twitter. So to quickly summarize, I'm not actually going to show any data from this, but we do indeed see that neuropilin is a co-receptor for NGS. And you might be able to tell that when you kind of get obsessed by a specific protein, there's a question of whether that would necessarily work in industry compared to academia. And I would point you, obviously, I don't know too much about industry, but I would point you to the other talks in this session, as well as this podcast, episode 14, where they actually talk about this specific idea. It's a really good podcast. I recommend listening. So very quickly, because I've run out of time, because obviously I'm doing the academic talks, so of course I have. So a few just key points. People ask me why didn't I want to stay in the US? So obviously I've come back again. I've set up a lab in Nottingham. People were saying you could do a K99, you've done 3 times as much. Your startup would be massive. Why wouldn't you want to stay in the US? So for me, in New York especially, almost everyone I worked with was international. And I felt very lucky that I actually had the luxury of picking. So I could come back to family and friends. I could have an option for parental leave compared to New York. But I could still do the science that I love. I could still look at the data in the same way and do the same kind of experiments that I wanted to do. So I basically had the luxury of getting to have that choice. So now I'm 6 months into this position. We've set up a lab and it's honestly so much fun. I know obviously there's a lot of tasks that you didn't even know that you'd have to do, but that's part of the fun of it, isn't it? And getting to set up that lab. I'm also going to mention that person that I said I met in the UK. We eventually managed to see each other after a year of long distance. We did have to meet in Mexico because of the travel ban, but that's fun. And then we ended up moving back here together. So the importance of those people in your life. So finally, final slide. Just a few top tips. So firstly, don't compare yourself to others because you have no idea how many failed applications they've had or papers that haven't been published, etc. Secondly, do the science and the job that you find fun. So I followed the academic path, but mainly because the part of the job that I love are things like writing, teaching, presenting, things like that. So do the job that you find fun. If you love being at the bench and if you hate writing, then listen to that feeling. And also, when it comes to comparing yourself, I've included this tweet in the background because I love this tweet, because it basically, what I took from it is that we need someone who loves every type of science in there. So this one in particular, some people make the big leap, some people fill in the details, some people are always right, some people aren't always right, but they change their mind. We need everyone who's good at different things to come together in science, academic or industry or otherwise. I'm a planner. Things don't always go to plan, but you can learn from each time it doesn't go to plan. And also really mirroring what Nikki said, it's the importance of who is there in your scientific journey, cheering you on, challenging you, etc. The people you just see day to day. So the people in Nottingham, the amazing people I met in New York as well. It really makes a difference who is there. So I'll leave it on this slide. I don't know if I have time for questions, but thank you very much.
Thank you, Chloe, for that incredible talk. We do have time for one question. There's a question in the Q&A. So this person asks, well, firstly, they say you've a very interesting talk, and they ask, how have you managed to get so many scholarships? Have you got any tips you can share?
Just keep trying. I don't know. What are the tips that I could share? I would say, in terms of actually applying for things, always read the question, which is always one of those annoying things that people say. But if they've asked 3 questions about, say, why you want to work in that specific institution, blah, 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 don't necessarily cut and paste from another application. Really write it for what you're writing it for. Also, listen to feedback. Listen to feedback, but with a pinch of salt. So sometimes you might receive feedback, say, on your proposal, and you might not agree with it. You might think, I can perfectly defend why I've done that, and I think I disagree with your point. So listen to feedback with a pinch of salt. But yeah, you will always get rejections. That's part of academia as well, isn't it? Is it 10% success rate for grants? So you're going to apply for 10 grants and you're probably not going to get 9 of them. So just keep trying. Celebrate when you submit as well. Don't celebrate when you get it, because then you get 10 celebrations rather than 1.
Thank you, Chloe. And if anyone has further questions, please save those for the panel discussion. Next up, Eddie will introduce our next speaker.
Thank you, Sam, and thank you very much, Chloe, for that fantastic talk. So our next speaker for today is Dr. Mark Suave. So Mark is a senior scientist at OMass Therapeutics, and the title of his talk is The Journey So Far as a Not So an Early Career Researcher. Over to you, Mark.
Thanks very much, Eddie, and thank you to the organisers for inviting me and the rest of the panelists for such a fantastic session. Yeah, so I thought I'd caveat this slightly, because in wanting to stay as youthful as ever, I always think of myself as an early career researcher. However, alas, you can't forever be in your early career. And I think technically, as the definition goes, I have since lapsed out of my early career. So I may be a bit of a fraud on this panel, but I perhaps can provide a bit of perspectives on there. So I thought I'd be upfront and honest with this, as a has-been and an old bogey on the panel. So like everybody else, I thought I'd start with a bit of my background. So I did a bachelor's in cell biology up in Durham, the northeast of the UK, well, of England. I then moved down to Imperial College to do an MRes in biomedical research. The reason being, I enjoyed the practical side of the lab in my undergrad project, but we only had a 5-week placement, really not enough time to understand if it was something I really wanted to do for my career. I enjoyed it, but in that master's year, I did 2 6-month projects, both lab-based. I found a deep love for being physically in the lab, discovering, even at the master's level, discovering new things and writing up about it as well. So it's from there that my love of biomedical research, and specifically getting into the kind of drug discovery, the pharmacology side of things, really took off. So it's from there that I took on a PhD in Nottingham, that I'll be speaking a little bit about now. So my PhD was in molecular pharmacology under the supervision of Professor Jeanette Woolard and Professor Stephen Hill. You can see their lovely pictures there on the right-hand side. And I have a lot of my career to date so far to thank. I have a lot to thank them for everything, for their mentorship, for their guidance, not only for my PhD, but all the way up to today and hopefully way into the future as well. So my PhD was investigating the pharmacology of an antibody. Now, this antibody was raised against the GPCR, the beta-1 adrenoceptor, and this interplay between biologics of antibodies or small antibody fragments and membrane proteins like GPCRs is really something that at the time I was doing my PhD was quite an interesting avenue to pursue and still is, but it's really exciting to be involved at cutting edge research. I should say as well that this PhD was part funded by what was then called Heptares Therapeutics, now Sosei Heptares, and some associated work from my PhD actually resulted in my first publication, which I'm proud to say was in PR&P. So represent PR&P in 2016, I think it was issue 4. So fantastic to be coming back and celebrating this 10-year anniversary here. In the spirit of Chloe's talk of being open and honest in that Instagram versus reality side of things, I thought I'd share a story from my PhD. In my experience, I think it would be really dull and boring if a PhD just went exactly to plan. I don't know of any PhDs that actually technically went exactly to plan from friends and colleagues. So it's those kind of pivot points, those bumps in the road, if you like, that provides a challenge, that provides something for you to overcome, that make it worthwhile and make it enjoyable and provide learning and growth through it. I'll just show some brief data just to explain this story really. So in characterizing this antibody against the beta-1 adrenoceptor, as a molecular pharmacologist, there are several types of experiments and assays you can do to understand how it binds, the functional responses, how specific the antibody is in this case, really treating it like a small molecule antagonist in a way. So here we've just got a concentration response curve at the beta-1 adrenoceptor with an agonist in the presence of increasing concentrations of antagonist. So a classic Shield style experiment, and you could see a nice rightward shift of the the concentration response showing competitive antagonism. So when I excitedly, in my first year, got my hands on this antibody and performed the same experiment, we were expecting this antibody to bind to the beta-1 receptor and be able to block agonist binding. So you would expect nice rightward shifts of these curves, but you can see here that the curves overlay almost exactly on top of each other, suggesting that there is no interaction in this case between this agonist and in this case the MAB, the monoclonal antibody. When I did the same experiment against the turkey beta-1 receptor, stay with me here, I'll explain why turkey in a minute. You can see that you get a rightward shift. So it wasn't doing anything at the human receptor, but when you did it at the turkey receptor, you actually got a kind of an experiment that you were expecting at the beginning. So this immediately showed two things that could have happened. One, this is a real effect, and that this was a true pharmacology of the turkey. Or two, I'd done something wrong and completely forgotten or done something erroneously in this assay. So after proving again and again that this was a real effect at the human beta-1 adrenoceptor, we then went on to interrogate this. And this was one of those pivot points in my PhD. We worked out that the antibody was raised against this peptide sequence down here on the bottom left, which represents the second extracellular loop of the turkey beta-1 receptor. And those residues that are highlighted in different colors represent residues which are different between turkey and human. When you do an experiment looking for binding in the middle here, middle bottom graph, you can see that with the antibody in the presence of the turkey extracellular loop peptide, you get a reduction in our absorbance here relative to the antibody by itself. However, when you do the same experiment in the presence of the human extracellular loop on the bottom right, you actually got a slight increase or no change in the antibody binding. And this suggested to us that the antibody was really specific for the turkey extracellular loop, but couldn't bind at all to the human extracellular loop. which backed up the functional responses that we saw in the top middle graph. I then went on to do a series of different tests and analysis. This is just looking at whole cell binding. So you can see again in the top left, you get in the filled circles, you get a reduction in specific binding of a radioligand in the presence of our antibody. It does absolutely nothing in the human beta-1 in the middle, comparing to just a normal small molecule antagonist in the open circles. And then if you mutated the turkey receptor to have the human extracellular loop, you actually got the human beta-1 receptor pharmacology. Again, further building up the evidence case that it was selected for the turkey ECL2. I then went on to do a series of experiments looking at single point mutations to try and really narrow down what the difference was. It turns out that it was this group of 3 residues up here in this DPQ in the turkey sequence that the antibody was really selected for. And so this meant that we had a really great tool for studying the pharmacology of this turkey receptor rather than the human receptor. And it really showed an inflection point in my PhD and a way that we can then think about, okay, well, how do we then pivot this to study a bit more the interaction between a biologic and a qPCR? Now we know this tool, now we know that it's actually a real result. We were then able to do a series of experiments and showed that it was conformationally selective as well, and produced a reduction in antagonist secondary messenger cyclic AMP without changing radioligand binding affinity as well. And I'm pleased to say, after lots of work, this ended up being published as well. So it just goes to show, if your first experiment, you think it may be a dud, it may be a failure, it may not necessarily be something that you've done, that may be a real effect, and don't always get disheartened by any failures. Any failure is just another door for an opportunity. So following on from my PhD, I then was able to apply and was successful in a postdoctoral position, staying on at the University of Nottingham. So again, this was around in the same group. So in Stephen Hill's group, working with Stephen Bridden, Professor Barry Kellum, and Professor Woolard. Again, you can see their pictures on the right. And this is really using the latest molecular pharmacology techniques to interrogate a variety of Class A qPCRs. We use techniques such as TR-FRET, BRET, and fluorescent ligands to really probe different Class A qPCR pharmacology. One thing that I was developing as well through collaboration with the Vrije University in Amsterdam in Professor Martin Smits' group was the use of nanobodies. So these are small antibody fragments to target one of these Class A qPCRs, CXCR4, and using this nanobody small antibody fragment to monitor allosteric interactions. So really kind of cutting edge qPCR pharmacology, but still using that biologic approach. One thing this really showed me as well was that it used a team science mentality. Working as part of a larger grant as well meant that we could achieve more by dividing and conquering the work and working together. And it was a really fun time. I had a great time working with so many wonderful colleagues, and it was just a real highlight of my time there. I should say publications thankfully came, which was wonderful, but there was a lead time. It took about 2.5 to 3 years before for the publications from this work that I was doing here before it's really paid off to fruition. So don't always think if you're embarking on something new that results will immediately happen. You've got to work at it, but they will come, I'm sure. This here just shows a variety of different techniques in kind of graphical form, if you like. So on the left here, we've got nanobodies. I was using the experience that the research group had with nanobrets and split nanoluciferase, some of the work that that Nikki was also talking about as well, and using new ways that we can target nanobodies, biologics, to interrogate this in novel ways to find conformational selectivity. We also then developed the technology and worked with Promega as well to look at internalization of cell surface, in this case adenosine A1, and together with Professor Kellum's group as well, talented postdocs and PhD students, used and developed more fluorescent ligands to target specific qPCRs as well. Through not only the work, but also collaboration with the BPS, with LRIG, with Adherent Rise. So this is a cost action group from the EU. And of course, with the Centre of Membrane Proteins and Receptors. So this is a centre between universities of Nottingham and Birmingham. You're able to collaborate more, and certainly in any walk of life in science, it is not, you are not a lone shark, you are not a single figure, a lighthouse. You are part of a wider team, whether you know it or not, and it's through that collaboration that you are able to achieve so much more. So it's really, all of this work couldn't have been done without any of these collaborations. What these collaborations mean is that you're able to travel, you're able to present your work, as Chloe was saying, and it's wonderful to go and experience new things, new opportunities, and it's just great to have fun. Now, all of these chances lead to collaborations, I was saying, and fun things here. Now, it's really exciting. And when I was getting to this was kind of the end of 2019, 2020. We all know what happened in 2020. It's a very common theme across the talks. This meant that we had to then pivot onto an online world, and it was wonderful to be part of so many different conferences and presentations, including ones that myself and my colleague Laura Kilpatrick set up as well on the behalf of Compare. But this for me represented a time of inflection as well. I was coming to the end of my postdoc. I had real life situations of mortgages and bills to pay, and didn't have a secure income prospect job looking at the end of my postdoc. So it was at this that I decided to look into other opportunities, and one of these opportunities was a career at OMass Therapeutics. So I won't go into this because I know that Nikki touched upon this as well in her talk as well. So OMass is a biotech now based solely in Oxford, originally formed as a spin out from Professor Carol Robinson's research group. And we are a biotech that's looking to develop our own drug candidates as well, using novel mass spectrometry and other pharmacological approaches. In my current role now, I am a senior scientist here in pharmacology. I have various roles and responsibilities, obviously lab-based, performing experiments, analyzing the data, working as part of a multidisciplinary team, so that collaboration is still very much present. One thing that I found really, really rewarding is, as I've gone from a PhD postdoc, performing the basic research, that fundamental research that's so important, I now am slightly more involved on the drug discovery and development process in that development side of things. And it's been wonderful hearing about those and learning all about the processes of how drugs go from a proof of concept all the way through to an FDA approved drug. It's a wonderful road, and I'm really enjoying and learning a lot. One thing to do is team science. I won't go into this in too much detail in the interest of time. Happy to answer questions. It's a wonderful thing that we did at Compare, and we're still doing at Compare. It's been recognized by the Academy of Medical Sciences, highlighting that collaboration, multidisciplinary approach. Volunteering as well. I would encourage you to volunteer for societies, for organizations. I myself am a volunteer for a group. We provide scientific content for free. We have so many different events and opportunities for you as early career members. And I would encourage you to come along if this is of any interest. We've worked closely with the BPS as well previously. I will just leave on this final slide showing opportunities as well. I encourage you to take every opportunity life throws at you, just in life in general, but certainly in your career. I think there are so many things that you can achieve if you just say yes and put yourself out there a bit. If you don't try, you'll never know how far you can go. And thank you again to all the organizers and the BPS PR&P for this wonderful opportunity. And with that, I'll finish.
Thank you very much, Mark. That's a very nice talk. So there's a few questions in the chat. How did you increase your visibility and exposure through actions at networking events such as conferences, poster symposiums? And how did that sort of help you stay connected with people in your field?
Yeah, great question. I think the honest answer is just going up and talking to people. It's advice that both Professor Willard and Professor Hill gave me was be bold and, when going to conferences as a PhD student, go up and talk to people. Don't be afraid, and just try and introduce yourself and keep that personal connection as well. If you're afraid to go in and feel like you might look a bit foolish speaking about somebody's work, try and make a personal connection with them first and then develop it into their work. And then from there, you develop these relationships over time, either, digitally by email, Twitter, whatever, or, from follow-up conferences as well. Just keep building on those relationships. And that's, to be honest, I would say that's the best thing you can do for visibility.
Perfect. And perhaps just a quick one. I was thinking of, while watching your talk, what's your advice for when your experiments just aren't working as you expect?
Yeah, great question. And I'm sure more people than would care to admit could relate to that. My advice would be, don't hide anything. Be as thorough and as rigorous as you can to ensure that what you're seeing is a true result. Obviously, troubleshoot it if it's something technical that you can work on. But look to your mentors, look to your supervisors, your colleagues, your friends for advice as well. I'm sure they will have and be willing to offer advice. And if it is a true effect, then try and find a workaround. And if not, know when to drop a particular line of questioning as well. That's as important to know when it may be time to change tactic as well. But keep persevering and know when it's a good time to stop.
Very good. Thank you very much, Mark. Over to Sam now to introduce our next speaker.
Thank you, Eddie. So our next speaker is Ashia Wheeler Crawford. She's a Wellcome Trust funded PhD student at the University of Nottingham, and her talk is titled From Jamaica to the UK, My Journey in Balancing Family and Research.
Hello, everyone. Thank you for joining us today. Let me see if I can get my slides shared with you. There we go. And we can all see that. Thank you guys for joining us today. And congratulations to PR&P on their 10-year anniversary. And thank you to BPS for supporting this webinar. So today I'll just be talking to you about my journey and how it is that I got to research and how did I do that, balancing that with my family. So I'm just going to start from the beginning where it all started. So for me, my journey started across the Atlantic in Jamaica, where I grew up. Of course, Jamaica is known for sun, sea, and sand. I didn't grow up anywhere near that. I grew up in a small farming community where, attending university was not necessarily the norm. But I was a child to a teacher, and her advice to me was always that education is the key to success. And she kind of instilled that in to me and my siblings. And we all, sort of took that to heart, and we all kind of tried to excel at education. And at the time, I actually really enjoyed going to school. And so, for me, it wasn't necessarily anything strange. So from there, I think my love for science really started in high school. So I went to Glenmuir High, which is, I think, where my love of science started. I had amazing teachers, and I think my biology teacher was a mentor to me. She believed in me. She gave me so much advice. She, allowed me to, to excel. And I think there is where my love for biology specifically, started to grow. I excelled in biology, got national awards for biology. And I think it was because of her mentorship and her guidance. And that was kind of very important to me. So because of my love of biology, I went on to study biotechnology. at the University of the West Indies. And here's just like a general education and career timeline. So I went to the University of West Indies in 2008 and did biotechnology. Following that, I actually, at the time, wanted to do a PhD. However, for some reason, after I applied, I did not hear back from the university until I knew I needed to do something. So I got a job as a biology teacher teaching A-level. And on my first day of the job, I got a call from the university saying, how comes I have not shown up for my PhD? So I found it was really, okay, we did accept you for some reason. We didn't send the email out. So that was, so on my first day of the job, I was like, should I quit this job and go and pursue this PhD? It was, quite a challenging decision for me. But at the time, I think I chose to stay with the job because I actually felt really bad just leaving the job on the first day. And I think that 1 year of teaching, I was like, okay, I'll do this for a year and then go back to the PhD. And 1 year became 6, and I stayed there. But, the idea of studying science and doing research kind of never left me. And during that time, I kind of then went on to do my MSc, eventually work in industry, and then now becoming a PhD student. So I'll go a little bit more in detail from being in Jamaica as a biology teacher. How did I make that decision to leave Jamaica and come to the UK and actually pursue a PhD eventually? So my decision to leave teaching, it was, again, it wasn't necessarily, I enjoyed it, but it wasn't necessarily the career path that I initially thought of. And I think sometimes you kind of get stuck in a rut. And I was like, this is not where I want to be, but how can I get to the next step? And I thought, okay, let me apply for master's. So that would be kind of the first stage in kind of getting back into the scientific field. I looked into doing masters at my University of West Indies. And then I also thought, okay, what about, going abroad for a master's? So I looked at masters in America, I looked in Canada, and also in the UK. And I think in the end, I settled on the UK because I think a lot of the MSCs I saw in the US were 2 years, and in the UK, they were 1 year. In America, they wanted to do these graduate record exam, which is the graduate version of SATs. And I'm not a big fan of these types of exams. So I thought, okay, I'd focus on the UK. So then the University of Nottingham, I found, it had a large international community. It was quite a research intensive university. And most importantly, it had scholarships for international students. Of course, I wouldn't have been able to leave Jamaica and come here and pay, outright fully for masters. So I received this Developing Solutions scholarship, and I think it covered 50% of my tuition, and that kind of helped me to be able to do that. So as part of my master's, we did a 3-month placement, and that placement I did at Nemora Pharma, which is a small pharmaceutical company based out of the Technology and Innovation Center at the University of Loughborough. And during that placement, it was quite interesting. I mean, the placement itself was not actually lab-based. It was, they wanted me to, do like a market survey of the vaccine market because the company wanted to go into developing vaccines. And at the end of the placement, they offered me a job. And the job was quite interesting. We were looking at formulation science. So less of drug pharmacology, but looking at how we could pair current and existing drugs with the proprietary microneedle technology that the company was developing. So our task was to develop new formulations, ensuring that they were safe, do stability studies, drug permeation studies. And at the time, it looked quite interesting, and I agreed. And also, as an international student, things we have to think about is like, okay, visa sponsorship. Okay, do I not take the job if I wait and my visa expires? So at the time, I thought the job was interesting, what they were doing, and they offered a visa sponsorship. And so during that time, I spent, I think, 5 years there. During that time, I, met my husband and we got married, started a family. But I think in that job, I still didn't feel fulfilled. It wasn't the field I wanted to be in. I like biology more than I do chemistry. So I was like, I think it was time for me to take the next step. So I decided to start either looking for a job, looking for another opportunity. And I think we had a placement student who was doing her PhD, and she did a 3-month placement with us. And I think she motivated me to apply for a PhD. I think she was at the time a single mom. She had 3 kids, and I was like, and she was doing an amazing job with her work as well. And I thought, oh, I can also do this. There are other people who are also having family and also doing PhDs. It's not impossible. So I thought, okay, PhD is the next step if I actually want to switch careers, if I want to actually get into research. I think doing a PhD is the first step. And so I went ahead and I applied for a PhD. I think with a family, I kind of had quite a lot of criteria. Of course, firstly, I was still would be classified as an international student, so I needed to find a PhD that would have funding for international students. I had a family, so the location was quite important. It needed to be within the Midlands, whether East or West Midlands, so it'd be travelable from where I was currently living. And of course, I wanted a project that was kind of focused on the techniques I wanted to learn, more cell culture based, and a supportive team. So I saw the drug discovery and team science program being advertised, and it was being funded by the Wellcome Trust. And when I looked at all the criteria, I thought, oh, it seemed like such an amazing program. And I thought, this is so perfect. This is what I need to do next. This is what is going to take me to the next stage of my career. And I applied for the PhD. Luckily, I was able to be accepted. And of course, when we did the interviews and I met, the members of the team, it also solidified that, okay, this was the right decision for me. This is what I want to do for my next step. And the project I selected was with the hypoxia and acidosis research group, again, because they were doing, all the techniques that I wanted to learn. So once I accepted this PhD and I started the PhD, then of course I had to, add something to it. So I decided during my second year that I was going to have a PhD in the middle of, have a baby in the middle of my PhD. Seemed like quite a strange decision, but again, there was a lot of things I had to consider. It wasn't an easy decision for me or my family, but at the time there were many things I had to consider. What were the goals that my husband and I had for our family? What did we discuss before I even accept this PhD? What were my personal goals? How would this affect my PhD? How would it affect when I would end my PhD? Again, I also had to think about my age. I didn't just go from undergrad to PhD, quite like a lot of the members of my lab. My journey was a bit more complicated. And so, I was a bit older than the people in my lab. And so I had to think about, how old I was at the time, how old I'll be at the end of my PhD. And so, that was quite important for me to not, have a baby when I was much older. I also had to think about, what were the financial implications. We had a mortgage, I'm doing a PhD. It's not it's not a job where you get in a high salary. What were the financial implications of adding another member to our family? And what was my support system like? Whether it's my support system in the lab, how would my supervisors feel? Would they be disappointed? And my support system at home, because with a family, it's not easy to just do things on your own. So all of these things, we kind of considered. And I also, spoke to my supervisors, spoke to the DTP, and they were quite supportive of my decision, that, if this is what I wanted to do, if I wanted to do a PhD and extend my family, they would do everything that they can to support me. And I think that, kind of made my decision much easier. So I decided to take that plunge, took the time off, and now I am back doing my PhD and in my 3rd year and making progress. So that was amazing. So just a bit now about my actual PhD work and what is that I'm doing. So my current research, I'm looking at the role of bromodomain and extra-terminal proteins in triple-negative breast cancer and angiogenesis. So at the moment, I'm focusing on triple-negative breast cancer because it is the subtype of breast cancer which is most aggressive. It occurs mostly in younger women, and it usually has the worst prognosis of all the subtypes. BET proteins are a family of bromodomain proteins. They have these bromodomains that allow them to bind to acetylated histone. So when they bind to histones on DNA, they basically regulate the transcription of a large number of proteins, including those that are regulated in hypoxia. And one of those proteins that are inhibited when you inhibit the BET proteins is VEGF. And VEGF is one of the key regulators of hypoxia, which is one of the key regulators of angiogenesis, which is of focus in my project. So we know that if you inhibit the BET proteins, you inhibit VEGF and therefore inhibit angiogenesis. But a lot of the drugs, so for example, BET-I, which is on the market, they target the entire family of BET proteins, BRD2, 3, 4, and BRDT. So my project is not quite pharmacology based because they're not drugs that target these all of the BET proteins individually. So we want to understand which of the BET proteins is most important and which ones should be potential targets. So instead of using drug targets, we're using CRISPR knockout system in order to study these BET proteins individually and try to identify which of them is an important drug target. So we generated these CRISPR knockouts, and with the CRISPR knockouts, we were able to show that it was BRD2 was quite important in the growth of tumor spheroids. So when BRD2 was knocked out, we saw a significant reduction in the tumor growth compared to when we knocked out BRD3 or BRD4. And then from that, we wanted to find out, okay, even though BRD3 and 4 weren't important in tumor growth, do they affect angiogenesis? Do they regulate genes that are important in angiogenesis? So we did this cytokine array. We tried to, again, we used the knockout system. We tested the conditioned media generated by these cancer cells, and we wanted to identify which cytokines, which were important in angiogenesis, were being regulated. And we saw that all 3 of the BET proteins were individually regulating VEGF, which again is a key cytokine in angiogenesis. They were also regulating other cytokines such as this urokinase plasminogen activator. So at the minute, we know that the BET proteins, they can individually regulate the secretion of angiogenic cytokines. And so now we need to be able to test this. Does this cytokine affect the actual angiogenic profile of the endothelial cells? And that's kind of what we're in the process of testing. So they all regulate the VEGF and they regulate a number of different cytokines, but does this actually translate into a change in the phenotype of the endothelial cells? And that's the next step that we're looking to find out. And so I would like to thank my supervisors the current and past members of the hypoxia group that I'm a part of, the Team Science and Drug Discovery DTP, Wellcome Trust for funding my project, and of course the BPS for supporting this webinar. And of course, last but not least, I have to thank my family, my husband for his continued support, and of course my kids for being as understanding as they can when I have to do very long experiments and be late in the lab. But you know, without them, I wouldn't be able to achieve what I'm doing now. But thank you very much. If you have any questions.
Thank you, Ashia. It's really interesting to hear about your story and how having a family in research is very possible. Unfortunately, we've ran out of time for questions. So if you have questions for Ashia, please hold them for the panel session. Eddie will now introduce our final speaker.
Thank you very much, and thank you, Ashia. That's fantastic. So our final speaker for today is Jess Coley from the University of Nottingham, and she is studying for a degree in neuroscience. The title for her talk is From Undergraduate into Research, Navigating the Transition. Hi, Jess, it's over to you.
Hi, Eddie, thank you. Is everything okay with my screen?
Yep, that's all good.
Okay, perfect. Hi everyone, I'm Jess. I'm an undergraduate at the University of Nottingham. Today I'm just going to talk a little bit about my background and my first steps I've been taking into research, kind of the whys and the things I've considered when deciding entering pharmacological research, and then kind of the skills I've gained. I just want to thank the Compare Lab where I'm currently completing my placement, the University of Nottingham where I'm doing my undergraduate degree, and then also the BPS for putting on this talk. I think it can be really scary to kind of think about postgraduate decisions or any big career decisions. So to kind of have a place to learn and talk about people's careers and experiences is really valuable. So just to go on a bit about my background and my story. So I actually started my first year undergrad in biochemistry. I really enjoyed it, but I found myself leaning more towards neuroscience modules. I really enjoyed learning about neuroanatomy, neurological disorders, and so I spoke to my tutors and kind of the heads of school and decided to actually transfer over to neuroscience, which was good. I didn't miss out on a year. I was in second year, but it was just something that I would have preferred doing. Then kind of the next big decision to make was due to the design of the neuroscience course. You can either do a 3 years bachelor's course or a 4-year course with an integrated master's, where the 3rd year is a placement year. So I've decided to do that, and that's where I currently am, completing my placement year in research. And interestingly, it's not in biochemistry or neuroscience, but actually pharmacology. And I just kind of like to get the message across that it's okay to change your mind and try things out to kind of realize what you like and what you want to do. Just as like a next step, the next kind of big decision that I'm thinking about is I'd like to see if I enjoy industry and kind of what's that like. So I'm applying for summer industrial placements for this summer. Prior to this year, I was definitely uncertain about what completing scientific research was all about. I had questions kind of what are the day-to-days of working in a lab, what the experience would be as a complete beginner, kind of what the career progression is, and also fundamentally, is it something that I would enjoy? So I thought kind of the best way to answer all these questions was to get stuck in and complete a placement year. So when kind of thinking about why I decided to do a placement year, it was really to answer these questions, to see if I enjoy working in a lab, to kind of spend a long time there and integrate myself within the team, and also build a toolkit of lab experience that I can then take forward into my career, and also explore pharmacology and see if I can then use the skills from that and neuroscience to use in my like next stages of research. So just kind of to know that it's okay to get stuck in and be passionate towards different things, because it can really help highlight your next career stages. So obviously I did biochemistry, I'm on a neuroscience degree, so why pharmacology? So I was introduced to pharmacology by completing a second year module. It was Dr. Nick Holliday's module. And I really enjoyed it. I thought it really helped kind of broaden my neuroscience knowledge, made the literature so much more accessible. And I also kind of enjoyed understanding like the rationale behind drug design and how that leads to different drug discoveries. And then I also liked understanding different pharmacological tools and then how that can be applied to understanding physiology and pathophysiology. The next kind of thing to consider was, do I want a placement in academia or do I want to get experience in industry? So I decided a placement in academia because I thought this would give me a wide range of experience and freedom in the lab, and it would enable me to kind of have trajectories and follow kind of paths in my lab experience and in my project. Also to be surrounded by people publishing papers, so it would improve my writing skills. Also be surrounded by PhDs, postdocs, and also PIs to learn from their experiences, see what they've done, and also see how I can apply that to my career. I also thought this would be helpful, kind of the things to consider like when applying. This isn't only directed at a placement, but also for kind of a master's course or for work experience or any kind of new lab. I definitely think one of the most important things is consider what you want to achieve within your time there. So read their published papers, kind of see the techniques that they're doing, see the pace they're working at, the people they're collaborating with, and see if kind of your ideas align. Definitely always apply as early as you can, but equally it's never too late to apply. I think there's always different teams, different groups that you can contact to see if you can get involved in that. It's especially on kind of an experience level. I also think probably one of the most important things is never be afraid not to approach people first. I think everyone's just kind of like an email away, and so especially kind of as an undergraduate, you're at a university where there's so many experts in the field where you can contact, see if you can get involved in what they're doing. And in fact, a little bit about my placement is that there wasn't a position in the Compare Lab for me originally. I emailed Nick Holliday, who I spoke about previously, who put me in contact with my now supervisors after a few interviews and a few chats, and we realised kind of how we'd want to spend the year. They opened up a position for me in the lab which wasn't there previously. So just to kind of note that if you show kind of like passion and that this is something that you want and this is something that you think that would help both parties, and also if you approach and knock on the doors, like something will open up for you, and it's a really good way of just getting your foot in the door and meeting new people. So just to tell you guys like a brief introduction just to my project this year. So we're investigating ligands that covalently transfer fluorescent cargo to the adenosine A3 receptor. I'm working in collaboration with PhD students and postdocs within Professor Steve Hill's lab, medicinal chemists at the University of Nottingham, and also early career researchers in the wider Compare lab. In terms of the experimental design, we're following similar approaches to Dr. Lee Stoddart's 2020 paper, which you can see on the right here, just applying fluorescent covalent transfer ligands to the A2A receptor, A2A adenosine receptor, and we're applying them similar approaches to the A3. Just to give you a little bit more information about what a covalent transfer ligand is. So it's made of 3 components. So here in purple, you can see the pharmacophore. So that's the selective part of the molecule. It has a high affinity to specific amino acid residues on your receptor of interest. So in my case, adenosine A3 receptor. Next, you have this reactive linker in the middle. So as you can see on this simplified mechanism, it's the part that undergoes the chemical reaction with the receptor, which leaves that detection moiety, in our case, fluorophore, attached to the receptor. And then the pharmacophore is then free to be released. And in terms of the use of these covalent transfer ligands, so it's really a pharmacological tool, which then can be used to study endogenous GPCRs. So that can be localization, functionality, and also orthosteric ligand binding, because the orthosteric pocket is free. Just to kind of tell you kind of all the different skills that I've learned. I'm halfway now, so I'm 5 months in, 5 months left. So I've been able to do a lot of molecular biology, such as PCR cloning, mutagenesis, and then using these constructs to be transfected into mammalian cells, either transiently or generating a stable cell line. Additionally, I've used kind of one of these constructs was a twin strep snap A3, which I then made a stable cell line of and then used to purify the A3 receptor, which you can see on the left in lane 6. And then that was ran using STS page. Also screened our covalent transfer ligands using molecular docking techniques and also nanobret. And then I've also done some confocal microscopy, which you can see on the right here. So that was using that twin strep snap A3 stable cell line, labeling that with an Alexa fluorophore and then fixing it, and then using the confocal microscopy to image internalization of the receptor upon application of the agonist, which in this case on the lower bottom right image, you can see it was NECA, and the white speckles here are showing the internalization. Just to summarize, kind of from making that decision of embarking my journey onto research, I've gained a plethora of experience, not only like lab-based, but also work-based. I've been able to meet and gain friendships with PhD students, postdocs, PIs, who are experts in the field and whom I can seek guidance and help from. Also, the placements enabled me to really improve my writing ability, which I can take forward into my final stage of my degree and my next career stages. It's also definitely in academia helped me gain independence in the lab, been able to troubleshoot, problem solve, and kind of design experiments. And I definitely think kind of a big take home message is just to how I've kind of gone about things is just to give things a go. And so I can now use my experience in biochemistry, my experience in neuroscience and pharmacology to kind of use that toolkit to take into the future. So I just want to thank everyone for listening, and I'm happy to take any questions.
Fantastic. Thank you very much, Jess. That was an awesome talk. So I've just got a question. So obviously you've just started your research career. You're 5 months in now, working as a lab scientist. What do you reckon the most important lesson you've learned from those 5 months in a lab has been?
I'd say really getting stuck in. I think not to shy and hold back. It's okay to be a beginner and it's okay to kind of not know things. So just to kind of ask everyone what they're doing, get involved in their projects, because everyone wants to talk about the work they're doing. And kind of, and it's just the best way to see what's going on in the field. I think sometimes as an undergrad, you can kind of be learning all the theory, but not know how it's applied. So just learn as much as you can and take every day as it comes and try and absorb all the information, I'd say.
Perfect. Well, thank you very much for your talk, Jess.
Thank you. I'll just share my screen.
Yeah. So we're now going to move on to the panel discussion. So we're going to invite back all of our 5 fantastic scientists back to talk and go through a few questions that you all have. So I'll just do a quick recap. So we had Nikki and her story going back and forth from academia to industry. Chloe and her story going back and forth from the UK and the US. Mark, who's had a varied journey around the UK with a stop in Nottingham for some turkey pharmacology. And Ashia, who's just got an incredible story, going from a teacher and then back to being a researcher. And finally, we have Jess, who's just given her talk on her promising start to her research career. So fantastic to have you all back, and let's get this panel session underway. So I think Sam is going to start with the first question. And for everyone at home, please ask lots and lots of questions in the Q&A chat for us to go over.
Great. So the first question I want to direct to all the panel members. So how has your career path changed from what you envisioned when you started out? And I'll go to Nikki to start with, I think.
Oh, sorry, can you repeat the question? I just saw one of the chat messages. I got distracted.
Oh, sorry.
So
how has your career path changed from what you envisioned when you first started out?
Yeah, I think originally when I was first exposed to academia, I kind of saw myself as a professor or the academic route. And a bit like what Chloe said, you have to kind of go with what excites you. And I found the industry side a lot more exciting and a lot more interesting. And I just had to kind of say to myself, like, this is for you. And I think when I finished my PhD and decided to do a kind of drug discovery postdoc, it was kind of best of both worlds. But at that point, I really knew, like, my next step would be, properly step into industry and see what I could do there.
Great. Ashia, do you have anything to add or any other expectations that you had when you started out?
I mean, I think for me, I wanted to be in academia. I think from my undergrad days, I think kind of the journey weaved into industry, into teaching because, you know, of opportunities that presented themselves. But, you know, I started this PhD because I saw myself, okay, I want to be in an academic lab. I want to kind of do this, as Nikki said, you know, be a professor. But I think now that I'm doing a PhD, I think my mind is starting to kind of waver a bit. You know, does it excite me enough, or do I think going back into industry, but now, you know, from a more biology side rather than kind of the chemistry drugs that I was working on, might be where I think I'm thinking of at the moment. So, you know, I'll see towards the end, but I think I don't know if that professor is where my dream is at anymore.
Interesting. Mark, how about you?
Yeah, interesting to get different perspectives, isn't it? I remember going into my interview with Jeanette, Professor Woolard, saying that I very much wanted to do my PhD and go straight into industry afterwards. And through doing my PhD, I discovered a deep love for basic research and how encouraging and then thrilling that could be. So I stayed on to do, and I pursued as much as I could down the academic route. Obviously, as I said in my presentation, COVID and reality of life and everything and bills, boring stuff, got in the way, which meant that I couldn't fully pursue that to the degree that I may have wanted to. But now I'm in industry, but I still love doing the basic research and finding out new things, but I'm loving where I am now. So it's changed, but who knows what the future has in store, right? It's you never want to close any doors, and yeah, it's certainly an interesting time. There's so much more back and forth, as you can see from the likes of Nikki and from Aisha's story as well. You know, there's so much back and forth cross disciplines and cross industries as well.
Yeah, absolutely, completely agree. Jess, do you have anything to add?
I definitely don't. I'm not 100% sure of kind of where I want to take it. I just know that I want to get as much experience as I can. So that's kind of, I think, getting as much as I can out of my academic placement, and then also seeing what it's like in industry and seeing kind of how the different layout is, what the different research is, and then kind of making my decision regarding like PhDs and stuff after that.
Great. No, it's okay to not know what you want just yet, as you can see from all the other journeys.
Yeah.
So, and Chloe, finally.
Yeah, so I think as soon as I did my PhD, then I kind of caught the bug for academia. It's not that I necessarily thought I would become a professor, because obviously it's really hard to get these positions. So at that point, I was kind of I had my option A and my option B, and I was trying to get my CV ready for both of them. Obviously, when I went to the US, part of me did think, is that going to change my decision of where I would want to work in academia? And obviously hearing from both sides kind of, yeah. So that I potentially was going to change my decision there, but then eventually, yeah, I made my decision to come back here. So.
Great, thank you. I think Eddie's got the next question.
Yes, I certainly do. So this one is for Nikki and Mark mostly. Do you think completing a PhD helped you transition into industry more easily than if you hadn't done one? Did it provide wider opportunities, or did you find it easier to apply for a wider range of roles? And there's also, I saw another question that was, do we think a PhD is sort of necessary to go into industry?
Right, well, I'll stop the awkward silence. So yes and no. There's nothing stopping you going from an undergrad straight to industry. You will go in at a lower position, but you can work your way up. What you'll find is if you want to transition from one company to another, that a lot of them will say, well, for this position, you need a PhD. And you can approach and say, well, I have 4 years working at this position and I have qualified myself. It's just, I think within companies, you can work your way up a bit better, but it is recognized as a certain level of a researcher. Whether you can gain that from experiences is definitely, you can definitely do that. And there's more to just a PhD than just the title. I think, yes, you can get extra opportunities from the contacts you make, but you learn a level of troubleshooting and resilience and things like that. So it is 4 years. It's not nothing to do. So it is quite a long time. And potentially in those 4 years, you could have worked your way up. But yeah, maybe Mark has something else to say.
Yeah, thanks, Nikki. No, I fully agree with everything that you said. It's more and more increasingly not necessary by any stretch. But from a personal level, it wasn't something, as you may have guessed from my previous answer, it wasn't the end goal. And I didn't do it to get into industry, to get to the position that I happen to be in now. It was more about the journey, as the personal journey that I wanted to go on, and making sure that I was doing it for the right reasons, I guess. So yeah, it's definitely not a requirement for entering into an industry, but it has certainly helped. And the contacts and the science and the skills that you've learned and that I've learned and developed during my PhD have definitely helped me, and probably the reason that I'm sitting here where I am right now.
Thank you very much. That's an awesome response. Sam, I think you've got the next question.
Yeah, so there is a question in the Q&A and it says, hi, I'm graduating this year and I wasn't able to find much luck with placements or internships. I'm interested in industry and neuropharmacology. Do you have any advice on what I can now do to find experience or a graduate scheme? And I think that's to everyone.
Yeah, I think for industry it's hard because like there are only so many positions and there is a limit. So what I could say is I know some people who go in it, for example, they can't get like a research scientist position. They might go in at a technician's position and wait for openings. I think that's also happens in academia. You might finish your PhD or undergrad and spend time working as a technician, and then they might progress to see where the openings are. But it's not, there is a tough market. Like, I don't think any of us interviewed at positions where there wasn't competition. So sorry, it's not really advice, but just see what kind of translatable jobs are available as well.
I can't say much from the industry perspective, but kind of mirroring what Jess said, there's also a lot of benefit to actually reaching out to people, say on LinkedIn, by email. And maybe it's a case where even just getting lab experience in an academic setting, just while you're waiting for that industry position to open, maybe that could still help because it will still build your CV and your technical skills.
Yeah, I'll just piggyback. Sorry. Oh, sorry, Aisha.
No, go ahead,
Oh,
Mark.
sorry, I was just going to say, and on top of that, attending things like this, other events that BPS and other learning societies are holding, that they're putting on and everything, but obviously for free in this case, it's a wonderful opportunity to meet people and ask questions, and you never know where those contacts may take you. Sorry, Aisha.
No, that's fine. No, I was just gonna add to what Chloe was saying. So, because when I did work in industry, you did have a lot of students. They weren't who, they could contact the CEO or, someone in the company, and they may be able to do like a 3-month placement or, a 6-month unpaid internship where you can gain those skills and that will help to build your CV. I did see that a number of students who kind of came through the company and did eventually move on to apply for PhDs just by getting like that lab experience that they could put on their CV and whatever the case is. So that's quite a good technique, either in an academic lab or even like a small company who they may not be able to pay a full intern, but if you're able to just work there for free for a few months, that can also help you.
Yeah, like I definitely recommend kind of maybe talking to like your supervisor from like your 3rd year project, see if they know anyone that you can get some experience of, just to kind of build it onto your CV, so then you're a bit more competitive in like industrial applications. Maybe that will help, and also put you in contact with a wider range of people.
Fantastic. Shall I go ahead with the next question? So probably it's the age-old question. What are the differences between academia and industry from your own personal experiences? Perhaps, Ashia, would you like to, because I know that, from your experience, well, you've got even more than just academia and industry. You've also got, schooling as well. So do you want to talk us through the differences you've found?
I mean, I think with industry it can kind of vary in terms of if you're in like a large company versus a small company. I think that kind of is quite different. So I don't have experience in a large pharmaceutical company, but for the company that I worked in, what I would say, because it was a smaller company, you kind of had your role was quite varied. So it means that you kind of gain experience in a lot of different areas. Like, I was interested in the formulation side, but because it was such a small company, I could learn, the analytical, the formulation. You kind of even had to kind of learn to write business plans to some extent. So it kind of varies. Whereas I think in a larger pharmaceutical company, I think your role is quite a bit more specific. It's like, okay, you're a formulation scientist and you kind of work in kind of a big team where each person has kind of quite a specified job role, which is a bit different I think in that small company that I work. But I feel like with academia, what makes it different for me is like, I think you have a bit more free reign on your project to kind of explore ideas and kind of think outside the box and kind of do something which is kind of important to you. In industry, I think they have sometimes a very specific goal, a very specific drug, a very specific outcome that they want, and you're kind of very focused on that. I think academia, if you're like really interested in basic science, if you're really interested in kind of exploring different ideas, and when you get to a point, you can like you can change the project in a way to kind of suit what's happening. I feel like I didn't have that much leeway in industry as much, so I felt like academia, it's good for like exploring and like learning basic science, and if that's what you like.
Oh, it's definitely, yeah. You're so right about the differences between a small academic setting, a small setting in industry where, for example, it might be like a university spin-off where actually the feel is very close to what you'll get in academia.
Yeah.
And you're so right that you can then do a lot more different roles compared to, working in a big industry, which I'd imagine is a lot different. Does anyone want to jump in on that?
All I was going to add, sorry, is just to kind of speak to if anybody is kind of concerned about, well, I'm at this point and I don't really want to be in academia and I don't really want to be in industry either, just to dispel the myth that it's not an either or situation, that the ecosystem, if you still want to stay in life sciences and drug discovery and anything relating to pharmacology, there is so much more out there than just those two camps. And I would encourage you to look widely, and whether it's patent law, whether it's sales, technical specialists, there are so many different career choices. So please, if you're sitting there thinking, I don't want either of those, please don't panic. There are so many ways that you can apply skills that you've learned in your undergrad, master's, PhD, postdoc, whatever school, that will be applicable to any career within life sciences. That was all I just wanted to say, Eddie. I agree completely with Ashia as well.
I'll just add one more thing on that. It's maybe a slightly more callous way of looking at it, but if you're comparing academia and industry, both are businesses, but where's the money come from to do the research? In academia, you get awarded grants. You may have to reach milestones to get the next lot of funding and everything. But in industry, you've been given money, but you're supposed to deliver. So if it doesn't look like it's going right, it's going to get cut. So the pace of the work in industry is much faster. If they want to look at something, they'll look at it quickly and efficiently. And I guess the one thing about the kind of freedom is you might have like a pet receptor that you're really interested in, but your skill set doesn't really cover the work that needs to be done. So you have to be kind of humble enough to go, I'm going to keep up to date. I'm going to go to the meetings, going to listen in as much as possible. But I think industry look much more at how can they make this progress. Whereas in academia, you might have more of a chance to develop the skills on the job.
Yeah, most definitely. So I think, Sam, we've probably got time for just one quick question.
Yeah, so in the Q&A, there's a question asking, what do you think are the criteria for someone who wants to study a master's in medical pharmacology? And I just want to piggyback on that and ask, what do you think the skills might be required for a PhD on top of that? And I think maybe if we just ask our panel members to give 1 or 2 skills each, starting with Chloe.
Yeah, so what is this for doing a master's or a PhD? Thirst for learning. That's going to be my one, if we're all going to contribute one.
Okay,
so that's reading papers and really looking into the data and really thinking.
Great. Mark's next on my screen.
I was hoping for a bit more time. I would say, yeah, Chloe's is a great one. I would say collaboration, working as part of a team. Even if it's not a big group, you'll still need to collaborate with others.
Yeah, great. Ashia? Sorry, you're on mute.
Sorry about that. I think collaboration is quite important as well, but I think independent thinking is also quite important, because you need to be able to, think analytically about the things that are there. You can't sit and wait and depend on everyone to kind of give you the ideas. You have to, also, be quite independent in your thinking and, come with ideas as well. Solutions. Be solutions-based. Don't just focus on the problems. Think about the solutions, I think, would be my advice.
Yeah, definitely. And Nikki?
Yeah, be flexible with how the work is going. You might have a very kind of clear view of what you want the project to be, but as Marcus said, things don't always go the way that you want them to. Data kind of comes out how it is, and you've got to be flexible and potentially be flexible and willing to pick up new skills you hadn't planned on using in the project.
And Jess?
I'd say probably be like confident and be keen to make sure that you can get the most out of it, and also collaborate as well with lots of different people.
Great, all brilliant answers. Thank you.
Yes, indeed. Thank you very much for all of our panelists for today. So I believe we now have an award that is going to be given out by Jen. Or Ross.
Hello, how are you?
Hi there. Good, thank you.
Yeah. What a fabulous, that was fabulous presentations, everybody. Thank you so very much for that. And I learned a lot as well, and wondered if that career advice would have been good for me. But that's very good. Were there any questions more for the journal for Ross or myself? Dr. Corridan's still awake at 3 a.m. Did you have anything, Dr. Rag, for us?
No, not particularly. Just that we're really excited and looking forward to working on that special edition with you both. And
Yes.
yeah, so do you have that award that you'd like to give out?
Yes, I do have that award. I'm delighted actually to announce we have an award from PR&P to celebrate our 10th anniversary. And that was based on the best paper by an early and mid-career researcher that was submitted as first author publication to the journal. And it was pretty competitive. But we did select a winner, and that's Dr. Sam Cooper from Nottingham. Highly deserved. And it's for the work on the beta-adrenoceptor agonist. So yeah, well done, Sam. Thank you so much for your excellent work, and to everybody who contributed, because yeah, that was a really fabulous paper. There's prize money actually. It's £1,000 to you, Dr. Cooper, that you can use for your research. So hopefully you'll use that to continue doing the great work that you're already doing. And yeah, thank you again very much, everybody, for your submissions to our journal, and look forward to future submissions. Thank you.
Oh, thank you very much for that award. I'm very pleased with it. Obviously, it was a team effort, so I think everybody that was on that team are really thankful for the recognition of all of their contributions to that article. So thank you very much.
Oh, well done. That's good. So we can probably close the meeting then, Dr. Rag.
Yep, absolutely. Yep. So I will just share my slides now. Okay, then hopefully everyone can see that. So just to finally wrap up then. So we've had a fantastic webinar, all thanks to the BPS, of course. So please, if you're not already a member, do consider becoming a member of the BPS. Undergraduates join for free. Early career membership, just £30 per year. And there's a huge amount that the BPS offer. We've got discounted rates of events and training workshops, access to study awards, prizes and grants, career development, and plenty more activities such as this one today. So just to finally wrap up, so thanks again to the BPS. There are some really great BPS events that are coming up. We've got Reflections and Innovations in Clinical Pharmacology. That's going to be held in London. We've got the 11th annual open meeting of the UK Pharmacogenomics and Strategic Medical Network, and that's also going to be held in London. And of course, the big one, the annual BPS meeting, Pharmacology 2024 in Harrogate, North Yorkshire on the 10th to the 12th of December. So finally, I'd just like to thank the speakers for their amazing talks today, and for everyone who has contributed from the audience for their questions, and everyone who's turned up for this. I think it's been a fantastic session, and I can't wait to see you soon at a BPS event. Thank you very much.
Thank you.